Today we're going to be finishing up the second half of our macromolecules lecture. So you should remember that we have four classes of macromolecules. We have carbohydrates, proteins, lipids, and nucleic acids. We've already talked about carbohydrates and lipids, so today we'll be discussing proteins and nucleic acids. So our goals and objectives are to describe the functions of proteins, to discuss the relationship of amino acids in proteins, to explain the four levels of protein organization, and to describe the structure and two types of nucleic acids of DNA and RNA. So we as humans we can be thought of as protein-making machines, and these proteins that we make, we make tons of them, and they have a lot of functions. So first thing to know, all enzymes are proteins, but not all proteins are enzymes. So we have certain proteins that act as digestive enzymes, and those would be amylase, lipase, pepsin, trypsin, and they aid in the digestion of food. So you can think about these enzymes. They would break down those covalent bonds and our disaccharides, all of our other macromolecules, and that's what's going to give us that energy that we need. So we also have proteins that help in transport. Probably the most common one we can think of is hemoglobin. That hemoglobin has albumin on it, and that helps the hemoglobin to carry oxygen throughout the blood and to carry lymph throughout the body. We also have proteins that help in structure. So we have actin, tubulin, keratin, and these make up the structure like the cytoskeleton. Proteins also play a big role in hormones, insulin, thyroxine, so these help coordinate our different body systems. They're also important in defense. They make up immunoglobulins, and that helps protect our body from foreign pathogens. They're also important in contractile or in movement. So we have actin and myosin, and these help control our muscle contraction. And they're also important in storage, and they provide nourishment in early development. So there's a total of 20 amino acids, and an amino acid will have a central carbon, which we see here. Here's our central carbon. We'll also have an amino group. We'll have a carboxyl group, like we see here, as well as a hydrogen atom. And then there will be some kind of side chain, and that's indicated here by our R. And our side chains, they come in different variations, but that's what determines which amino acid that we are dealing with. So whenever these amino acids, they come together to form a chain, and they make protein, but they are not themselves proteins. They don't become proteins until they are linked together by peptide bonds. So without that peptide bond, you don't have a protein, you just have an amino acid. So remember that all enzymes are proteins, but not all proteins are enzymes. So enzymes act as a catalyst, meaning that they speed up chemical reaction. So this is very important when it comes to our metabolism. And think of metabolism as all of the reactions going on throughout your body that help you to sustain life. Without enzymes, we couldn't survive. Also, enzymes can be used over and over again. However, they are specific to a certain job. We have a whole chapter on enzymes later where we'll discuss them in deeper detail. So let's break down the word polypeptides. Remember that poly means many, and peptides mean protein. So a polypeptide is just a large link of many proteins. Now, these proteins will consist of one or more polypeptides, and they fold to make a certain 3D shape. This shape helps determine what their function will be. Now, remember, proteins are very specific, so they will have only one job. They're not multitaskers. And then we have amino acids, and that consists of a central carbon, a hydrogen, an amino group, a carboxyl group, and an R group. And these are held together through peptide bonds. Now you should remember that sugars are held by glycosidic linkages, and that fats hold their glycerol and fatty acid tails together through ester linkages. These proteins, though, they are linked by peptide bonds. So to determine how many peptide bonds you would have in a protein, just take the number of amino acids and subtract 1, and that's how many peptide bonds you have. So for example, if you had 300 amino acids in a protein, then you would have 299 peptide bonds. Now, these peptide bonds are formed through another dehydration reaction. So you see here again, hydrogen and oxygen, hydrogen coming together, and we see our water leaving this reaction. So we have a dehydration reaction. And we see this peptide bond here between our carbonyl group and our nitrogen. We also see our amino end over here, our NH2. So protein shape determines function. We have four shapes, forgive me. We have a primary, secondary, tertiary, and quaternary. So our first is primary, and primary just has our carboxyl end over here, and we have our amino end. So this one is snake-shaped. It's really pretty simple. Just have the carboxyl end and the amino acid. So our secondary structure, that would have beta-pleated sheets and alpha helixes. And that's pretty much all there really is to that. For our tertiary structure, this will consist of both the alpha helixes as well as the beta-pleated sheets. However, these are held together by a disulfide bridge, and we see that over here. So that allows the structure to start folding on to itself. And then we have quaternary structure, and quaternary has 4 tertiaries put together. So we discussed hemoglobin a little bit earlier. Here is a hemoglobin molecule. We have alpha helixes, our beta-pleated sheets, and we have our 4 tertiary proteins that are joined together. Now, this hemoglobin molecule has an iron within each one of these subunits, and this iron will actually create an affinity for oxygen, so that oxygen wants to bind to this iron, and that's how we actually have oxygen go throughout our blood. So that could be, if you don't have enough iron, that could be an issue, and it could cause what's called anemia. Now, anemia is usually more commonly seen in females, and that's because females have their menstruation period. Oftentimes anemics are going to be women, and they'll be prescribed iron. It's not as common in men. In fact, men usually don't need to take iron because too much iron can actually cause some heart problems. So proteins they can be used over and over again unless they become denatured or if they're killed. So there's a couple of ways this can happen. If a protein overheats, if there's too high of a temperature, then those hydrogen bonds will come apart. Salt can be used in commercial buildings to kill fungus that's growing on the outside. pH too, if something's too acidic or too basic, that can kill proteins. And you can link that back to homeostasis. Remember that blood needs to be between a pH of 7.35 and 7.45. If it's outside of that range, then your hemoglobin isn't going to be able to function, and you're not going to be able to carry oxygen throughout your blood. So protein folding is assisted by chaperonin, and these proteins protect the polypeptide while it's being folded. So we have 2 different types of nucleic acids. We have DNA, or deoxyribonucleic acid, and RNA, which is ribonucleic acid. So DNA and RNA are very similar. The only structural difference is that there's 1 less oxygen on the sugar in DNA versus RNA. Also, they are both found in the nucleus of the cell, but as to where RNA can leave the nucleus, DNA will only be found within it. So if any reactions need to happen with the DNA, it's going to happen inside the nucleus. So our genes are made up of both types of nucleic acids. So both DNA and RNA make up our genes. And genes are units of inheritance. So they help with the storage and transmission of hereditary information. So nucleic acids have three components. They have to have a nucleotide, a sugar, and a phosphate. And this the phosphate group. And this sugar and phosphate are going to be joined by a phosphodiester linkage. So we see our phosphate group here. That's a terrible circle. There we go. Then we have our sugar, and then we have our nucleotides. And then we have our phosphodiester linkage here between our phosphate and our sugar. So DNA actually directs RNA synthesis, which directs protein synthesis. So again, let's go over our nucleotide. We know that we have a pentose sugar. Pent means 5, so we have a 5-ringed sugar. We have our phosphate group, and then we have a nitrogenous base or a nucleotide. So let's discuss the different types of nucleotides. So you will be responsible for knowing the difference between pyrimidines and purines, knowing the different names. So let's first off start with our pyrimidines. There's a nice little mnemonic that will help you to remember your pyrimidines. And your pyrimidines are these single ring sugars right here. So we're going to use the tool cut pi. All right, and then we see here that we have our cytosine, that's our C, uracil, and then thymine, and then pi for pyrimidine. And then what we have left over is our adenine and guanine, and these are our purines, and our purines are our 2-ringed nucleotides. So also, whenever we're talking about DNA, there are 4 bases that we need to know, and we need to know their complementary base. So in DNA, we have adenine, and that adenine is going to pair up with thymine. And then we have cytosine pairing with guanine. Now in our RNA, our thymine is replaced by uracil. So in RNA, our adenine is going to bond with uracil, and then our cytosine will still go with its complementary base of guanine. So DNA consists of 2 strands of nucleotides, and they come together in a double helix, and they're held together by hydrogen bonds. So again, we have 2 chains of nucleotides being held together by hydrogen bonds. So again, we have nitrogenous bases of our DNA. We've already talked about this, but we have our thymine matched up over here with our adenine. We have our hydrogen bonds between these nitrogenous bases. Let's go ahead and circle one of these bases. So again, we have a nitrogenous base. Then we have that connected to our sugar, and then we have our phosphate group. And between our sugar and our phosphate group is that diester linkage. And over here we have our hydrogen bonds. So again, just remember that our nitrogenous bases are joined together by hydrogen bonds, and that this is done so in a complementary fashion. Our adenine joined together with our thymine, and cytosine paired with guanine. So we talked about the functions of proteins. There's a lot of them that you'll need to know. We talked about the relationship of amino acids and proteins. Remember, amino acids make up proteins, but they're not a protein until a peptide bond is there. We talked about the different levels of protein organization. We have our primary, secondary, tertiary, quaternary. Remember that proteins can be reused, but they have specific functions. Also, we talked about the 2 different types of nucleic acids being DNA and RNA. You'll need to know the difference. DNA is double-stranded, RNA is single-stranded, and as to where DNA has thymine, RNA has uracil. All right, guys, I'll see you at the next lecture.